So I'm Eric Roberts. I'm with the California Environmental Health Tracking Program, which is a CDC-funded nationwide initiative. There's about 22 grantees nationwide, and with the cooperative agreement with the California Department of Public Health. That's what I do. And part of sort of the overarching thing with tracking is to develop data resources for both environmental hazard surveillance and health surveillance. And this leads to a lot of questions about what sort of research can be done, et cetera, and that's how you wind up with this project, which was really kind of done as a demonstration more than anything else within the context of tracking. It's obviously had a certain amount of legs. So I'm going to really briefly touch on autism pathophysiology such as we understand it, or rather don't understand it. And I think Tracy and Mike are going to be getting to this in a much more professional way later. This is really me, who pretty much just has a med school education, looking at the literature about where autism might come from. It's a disease that we define entirely clinically, so we don't really have any narrative to explain it, such as an infection or neoplasm or any of the basic pathology narratives we have. So this is my reading, and it's granted it's a little bit more of an amateur one. I would say that one category of pathophysiology is underconnectivity of the brain cells. That's sort of a theme that emerges when people look at abnormalities that people with autism have compared to their normal controls. People find fewer neuron connections in specific parts of the brain, both cortical and subcortical, particularly cerebellar, and pretty specific to GABA-mediated neurons. Okay, GABA is gamma-aminobutyric acid. It's a neurotransmitter, and it's intriguing because that's partly related to some of the inhibitory processes of the brain, which is interesting because autistic children very commonly have epilepsy. But another one is because GABA-mediated neurons play some fundamental roles in the synaptic patterning that happens during neurodevelopment. So it's a big teaser to have that finding. And then several of the genes related to autism govern the scaffolding that hold brain cells in position with respect to each other, and for the synaptic, so like when people talk about shank and the neurolignin and the neuroligand, and I can't pronounce those, that's what those are. Another one, people are noticing issues about systemic immune activation with autistic patients. Cellular and molecular measures like the immunoglobulin profile, like the cytokine production, those seem to be altered in a lot of autistic patients. An example of the overlap, one particular glial cells, for example, in the brain are chronically activated in autistic children. Well, those are immune cells, but they're also in charge of sort of holding all of the neurons in place through the relation to each other. So is that under number 2 or number 1? You decide. And then the other piece is endocrine disruption. People talk a lot about the sex differences in autism. And we know that particularly thyroid and estrogen sort of hormonal milieus are really important when it comes to neural patterning, both prenatally and postnatally. Human-made chemicals in the environment. These are figures that a lot of us probably have memorized because these are very well known. But in case you don't, there's more than 80,000 new chemical compounds that have been developed since World War II. Okay, these are all put into the environment in various forms. Obviously pesticides are put into the environment in certain circumstances that are very different than something that's actually actively exposed of. Of these, 3,000 of them are what we call the high volume chemicals, more than 1 million pounds annually. And about these, we know very little about what their toxicities are. We have certain protocols that a body like the EPA goes through to ascertain the safety, but with something as nebulous and poorly understood as autism, there's really no screening test that you could do. So, my bullet at the bottom. Given that, why am I picking on the pesticides? And that's a really good question. I want to underline that there's nothing special about the pesticides. Most of the ones that we talk about have relatives that are being produced that are not pesticides. So the organochlorines are related to the polychlorinated biphenyls, and they're related to hexachlorobenzene and all these other things. So other chemicals that are produced through industrial processes are also produced, are used in pesticides because that's what we use them for, but it's our definition of them, nobody else's. So, but why? Well, the issue really is that California is very unique. And Jeff alluded to this. The Department of Pesticide Regulation maintains a record of the date, location, quantity, ingredients used in every professional application of pesticides throughout the state every year. That's 1.7 million last time I counted every year. It's a huge amount of data. It's collected mostly at the county level and then sort of centralized with the state DPR. And to my knowledge, it's the only one. So thank you for doing that. I don't take it for granted that it'll be forever funded, and I don't understand sort of the ins and outs of how that works, and the state's in a lot of trouble. But it's a data source that really nobody else has on the planet. There are issues about it as far as it's very difficult to tell completeness. It's hard to tell how much people are complying with the regulations for reporting. It's hard to tell accuracy in terms of date and place that it's being done, but it's a pretty incredible resource when you think about it. Also at the Department of Public Health, there have been some real pioneers in terms of autism surveillance. That's not quite as unique as the pesticide use reports. There are a few around the country. The neat thing about California, and there are several around the world, the neat thing about California is that we do have a Department of Developmental Services, which is sort of a single state agency that is in charge of providing services to a very large number of autistic children and their families throughout the state. And so the folks at DPH have done a lot of studies on sensitivity and specificity of using those files for surveillance. And there's some really forward-looking people at DDS who have made the resources available to DPH so that they can collaborate. It's really rare. So what we have here is an opportunity to examine the spatial and temporal patterns of pesticide applications as well as autism cases and controls. Okay. And so there's a certain amount of looking for your keys where the lamppost is because there's light there as opposed to looking where there isn't any light. But I think it's a valid pursuit. I will add that when I talk about these pathophysiological processes, a very large number of the pesticides that we use have been implicated in these processes. An awful lot of them, not as their primary mode of action, but as sort of a secondary mode, not having to do with insects, do bind to GABA receptors. It's very common for like organophosphates and organochlorines both. A lot of them stimulate the immune system. Pyrethroids are really good immune modulators. And then several of them, and I haven't noticed a pattern of sort of which ones are and aren't, but there are lots of concerns about endocrine disruption for all of them. So this is not far-fetched in terms of theory. So I just said that we have these resources. What have we been doing with them? Well, the first thing we do is we take the DDS records and we link them with birth records, because the issue is we want to deduplicate them. We want to be able to sort of say, okay, of everybody who was born in this year, what percentage of them developed autism within a certain period of time. You can't do this until you actually link your birth records and your DDS records. California, like many states, has vital statistics that are very well developed. So this also means that we can have covariate data, like maternal and paternal demographic information, the address that mom had at the time that she gave birth, things like that. That might not necessarily be in the DDS records, but they will be vital statistics. Okay. So here's the study example that, sorry it took so long to get to it, that Jeff asked me to kick off with. There is generally concern in agricultural communities about residential proximity to pesticides, known as pesticide drift. That's the presence of measurable concentrations of a compound in locations beyond the site of application. often for extended periods afterwards. It's a documented phenomenon. The only difference is that if it stays on the location, then we call that occupational health. And if it drifts off into somebody's residence, then we call it environmental health. So it's a hypothesis-generating study. We use the DDS files. A little background there. It provides services without regard to citizenship or financial status. The referrals come from clinicians, schools, and families. And they use a DSM for diagnosis. And we included all of the autism spectrum disorders, so the autism, Asperger's syndrome, and pervasive developmental delay, ALS, not otherwise specified. So we match these to the birth records using child's first and last name, et cetera, et cetera, et cetera, for a 3-year period. The 3-year period. We had 465 when we sort of cleared out all our data quality issues for cases. We matched these with 15 full-term normal birth weight live birth controls. There's a potential confounder, since this is autism and we have some little idea of how it goes, if there could be another seasonal pattern like pollution or infectious agent that could have something to do with autism. Could that be a confounder? So we matched everybody by the date of mom's last menstrual period. So as far as time goes, everybody was in the womb at the same time when they're being compared to each other. So exposure data. So DPR, we had 6.7 million applications from 1995 to 1998. And it was 96 to 98 was our birth cohort, but we wanted to catch the period before that. As having gestation. Temporal resolution is day. People write down on their usage application what day they're going to do the application. And I think that piece is actually considered pretty accurate within a couple of days. The spatial resolution is actually the PLSS files, which are actually sort of 1 mile squares for most of the state, which isn't that accurate. The Department of Water Resources allows us to actually refine those to land use polygons. So in case you haven't figured out, this is sort of a data linkage mania, all right? We're taking the DDS client database and we're linking it to birth records using the address of and the dates of pregnancy. We're linking that to DPS pesticide use reports. And then based on the crop, we're linking that to the Department of Water Resources land use survey so we know exactly where in space the cornfield was or the cotton field or the apple orchard or anything else. So, and I'm going to have a quiz on this. So many, many compounds, time periods, and distances to fields considered. I won't talk about this too much because I don't think this is a group that really wants to go into biostatistics. But I could be wrong. The analytic emphasis becomes one of eliminating associations coming from multiple testing error, okay? So we did that. It's something that geneticists are very familiar with. The pesticides with associations remaining after the elimination of all of the multiple testing were the organochloranes, okay? That does not mean that there's no association with any of the other pesticides, but it means that the one that really stood out, given how we sliced the problem with the organochloranes. And I'm going to detail that just a little bit. The green bar up here, I hope you guys can see, is a timeline of pregnancy, okay? First trimester, second trimester, third trimester. The time of peak organ formation is right then, so mostly we call this the second and third trimesters the fetal period. The third trimester is the peak fetal growth, but the times that the tissues are actually getting organized in relation to each other is organogenesis, and that happens sort of in the middle of the first trimester. Okay. We had a variety of hypotheses. You can see those in purple time periods. Well, we thought, okay, the time leading up to about 21 days after fertilization, that's when the neural tube closes. That's when neural tube formation is. So we'll just say, okay, that's a hypothesis for when that would be important. And that's because there have been some studies, they're actually kind of old now, looking at known mutagens and realizing that actually the neural tube closure could be important with autism. The second one is central nervous system embryogenesis, and so we took the CNS organogenesis and we added a little bit of B time on there, so we tested that time. And then we also had sort of a blanket, relatively brainless hypothesis. Well, what if just sort of the amount of pesticides that you're exposed to during gestation could be related to autism risk? And so we tested that one too. And the one that we found was actually the central nervous system. All of the, when we went through all of these statistical tests, and we came up with, there were a few that just sort of stood out after you did all of the corrections for multiple testings with really tiny p-values. They were all organochlorines, and they were all exposures during the CNS embryogenesis period. Okay? And then we did something that is sort of cheating unless you state that you're doing it ahead of time, which is we looked a posteriori for when the greatest correlation was. Is there a time period where that we should have chosen if we were going to sort of be more prescient about it, rather than the CNS period? So this was the time that we found, and it's actually, it starts with the beginning of CNS organic embryogenesis and goes about 8 weeks later on. Okay. So, but that's a posteriori, and you kind of need to label that, otherwise you're sort of cherry picking your data. So. Table of results, just to make everyone's eyes glaze over. We're comparing to the people who were not exposed to any organochlorines within a 500-meter radius of their home. And then for non-zero, there's the first quartile, second quartile, third quartile, fourth quartile. For CNS and our a posteriori, you see that there are no significant associations until you get to the fourth quartile. For CNS, the odds ratio for, actually it's a relative risk, for autism pretty much quadrupled. For the a posterior, went up by about 7 to 8. Okay. I should mention we were controlling for some basic demographic variables such as mom's education, race, ethnicity. We also controlled for the regional center of DDS that actually did the diagnosis, just in case there was any diagnostic variability. Exposure misclassification is the big limitation to this kind of thing. People change their address during pregnancy. It's actually one of the most common times for people to move is during pregnancy. We also don't know how much time mom spent at home. So this is, she may have had that address at birth, but she may have been, commuting for another part of the state that entire time. So that's a big question. For all we know, we think that those are what we call it just entirely random misclassification errors, so that it shouldn't be biasing our effect estimates anyway, except worse than null, but we don't know that for sure. So there's little information available besides basic demographic variables, so we don't know if mom was taking folate, for example, so that she had to prevent neural tube defects or anything like that. And the children who are less severely affected by autism are going to be underrepresented because if they're only mildly affected, then they might not be getting DDS. The findings also hinge on a very small number of exposed children. There were 113 kids who were exposed, and so the highest quartile is just 29 kids, which is amazingly small. The odds ratios were so huge that it was significant anyway, and quite so. But it's the sort of thing that really begs to be followed up. And ideally, I would love to follow this up in another state or another country where we have completely different ascertainment and exposures. But guess what? I can't do that because California is the only place that has these kinds of resources. So other pesticides groups could be associated with autism risk, but they're not evident given the small sample size and given the fact that we really had to set our a priori choices of temporal periods up at the beginning of time. Our work since then, especially in the absence of doing another study, has really been on developing biostatistics so that we can not quite be as beholden to our a priori assumptions and kind of let the data sort of tell us when the temporal variability happens using a lot of hierarchical modeling, but that's a topic. Any questions? Yes.
What was the I didn't quite hear it. The temporal period. What was going on at that time exactly? You said it was a higher rate of stuff in the air. Could you explain a little bit?
So you're talking about our worries about temporal confounding. Okay.
No, what was going on? What made you choose the time period that you did to study?
Okay. I think you're latching on two things, but we wanted to test to see if not only living near the pesticide applications is important, but we wanted to know if it was important at a specific time, because that would be much bigger. So we needed to specify that time period up front. So we came up with these hypotheses. One of them, well, it would be the period leading up to neural tube closure. That was important. And this is me harking back to first year medical school embryology. Okay, nothing fancy. So we had one hypothesis about neural tube closure. We had another hypothesis about overall brain embryogenesis, and then one sort of for all of gestation. Okay. There was another question. We thought, okay, what if we do this and we have an association, and someone says, well, hey, all the pesticides are applied in the summer. That's when the air in the Central Valley is really bad. So what do you do with that? And so what we did is we made sure that all of the cases and controls were completely lined up. They all were conceived pretty much at the same time, and so they're all developing their brains in utero at the same time. So that's a epidemiological technique for getting rid of that compound.
Okay. And so were they in that period during that, like the summer months are worse, you're saying?
Well, you know, I don't recall exactly when the organochlorines are, and we were looking at all these different pesticides, so that was just an example.
Okay. Depends on the crops as well.
Yeah.
Right.
Okay.
I just want to make sure I understood your results table. Is the conclusion that there's a critical period in that first trimester where exposure would increase the risk of autism?
Yes.
Okay.
Yeah. Well, okay, yes, with caveats. We didn't measure exposure.
Yeah.
Technically speaking, that means you took a blood sample or something, or a urine sample, and found evidence of that in mom's body. So it's really...
So theoretically...
Yeah. Basically what we found is, the precise way to say it is residential proximity to organochlorine applications during that time seems to constitute a risk factor.
Yeah.
Two quick things. One is I'm making the assumption, I assume others are, that the geographic nucleus of where you are most likely to find this kind of result is in the Central Valley. Is that correct?
It is true. It's there's a couple things going on, and I'll answer briefly speaking. Yeah.
Okay.
It's definitely the Central Valley has some of the highest pesticide use in the world. I believe the San Joaquin Valley, which is sort of the lower half, uses about 20% pound per pound of the nation's pesticides. So this is a good place to study that. A piece of it, though, is sort of looking for your keys under the lamppost, in that when people do the pesticide reporting for urban functions, like they're spraying the right-of-way or a park, oftentimes they just sort of report for the entire city or the entire county. So we're not getting the same spatial resolution for non-agricultural applications. So we specifically chose the 19 counties in the Central Valley that seemed to us would be predominantly agricultural.
Okay, and just a couple other quick things. One is on the organochlorides that you've sort of fallen out here. Do we know much about the breadth and depth of their use in culture?
Thank you. When you say organochlorines in most environmental acting meetings, they mean DDT, and of course we don't, because we don't use DDT in California. In this case, it's two pesticides. Diclofol is one, which is almost DDT, but it's got a hydroxy moiety on it. The other one's endosulfan. They both have a much lower persistence in the environment, so it makes a little bit more sense that there would be a temporal association going on. They were used about 30,000 applications, if my memory serves, in the mid-90s, and have fallen to about, I think 5 or 10. I have this written down. So they're actually sort of on the way out for whatever reason, and I'm not entirely certain. So it's something that sometimes people ask me that because they're wondering, hey, could this be a reason why autism is going on? not in any simple way.
Just one other quick comment, and that is about the budget cuts as it relates to EDS and their ability to continue following and maintaining this kind of data flow for researchers. The state has pretty much decimated the Early Start program and knocked 17,000 children off eligibility. So it remains to be seen what the database, how that's going to be affected over time. In addition to that, EDS itself says that they serve about 20% of the kids that are on the spectrum of autism. So whatever we're looking at is a piece of the puzzle that is not the whole thing.
Right. Thank you. Do we have time?
I'll do one more and then we'll get through all the presentations. We should have a nice chunk of time for two, but go ahead, Lisa.
I'm just curious, it's well known, at least I was told in the last workshop, that autism has a very strong genetic basis.
Yes.
And I was wondering if, probably the data isn't comprehensive enough, but if you could analyze it in a way that would answer the question whether these increases are related to the genetics underlying all of this, meaning that if you're already genetically predisposed, is that when this hits? Or does this affect the entire population?
That would be great if we actually had a study where we could genotype the parents and the children and figure out what people's, maybe there's a subpopulation where this happens.
I mean, not necessarily genotype, but know the family history.
Sure, yeah. The only thing I've seen along that vein are, I think there was an Italian study where they looked at peroxidase, which is a detoxification enzyme for organophosphates. And they noticed that people who didn't detoxify as well, the ones that had that phenotype, they were more likely to have autism. So they said, it could be something like organophosphates in the atmosphere. I mean, that's a really indirect thinking. It's very important thinking. So. Okay, thank you.